urinary system, integumentary system, and so forth. You need to know examples of structures that sit within each, not details, just examples of structures and a generalized function of what each of these organ systems do and how they perform it. And then the last one for the very first lecture, which is Intro into AMP, is define homeostasis and the terms positive and negative feedback. All right, so positive and negative feedback is, firstly, homeostasis is, homeo means similar, stasis means standing or standing still. The body needs to maintain homeostasis. That means it needs to maintain a happy, healthy equilibrium of physiology, of function. What are these? Well, for example, we've got temperature. That's negative feedback for homeostasis. We've got labor. That's positive feedback for homeostasis. It all works via the same components. You have some sort of stimulus, which is picked up by a receptor. That then gets sent to the control center via an afferent signal. And the control center can be multiple organs. The major organ that's a control center is usually the brain. And what the control center does is it takes this afferent signal coming from the receptor and makes a decision as to what it wants to do with the signal, okay? So it evaluates it and decides and then sends an efferent signal that's away from the control center to the effector, and the effector is always going to have the effect. For example, with temperature, the stimulus can be a drop in temperature. This stimulates thermoreceptors. That sends an afferent signal via a nervous system to the brain, specifically the hypothalamus. That's the control center. It then evaluates this and says, hey, it's too cold. I need to warm the body up. So it sends an efferent signal to the muscles, and the muscles contract, relax, contract, relax, contract, relax. That's shivering, releasing heat, and then that brings the body temperature back up. And the muscles contracting are the effector. So you'll need to know examples of each of these homeostatic components for the end of trimester exam. All right, that's the end of the first lecture. The second lecture was going through body chemistry. So the first one says define and distinguish between the terms atom, element, and ion. What's the difference? So you've got the periodic table, right? You've got hydrogen, helium. So remember, you got hydrogen, helium, lithium, beryllium, boron, carbon, nitrogen, oxygen, fluorine, neon, sodium, magnesium. You've got a whole bunch of different atoms. Now, the atom on the periodic table says that if it's a hydrogen atom, it's going to have one proton, one electron, one neutron. Okay? Helium, 2 protons, 2 neutrons. Right? So this is what they're neutral. That's what these atoms are. Now, an element is a hydrogen element will be comprised of multiple hydrogen ions. That means hydrogen element is going to have multiple atoms that have 1 proton, 1 neutron, 1 electron, okay? A helium element is going to have multiple helium atoms, 2 protons, 2 neutrons, 2 electrons, all right? Now, that's the difference between atom and element. When we've got ion, what's an ion? Well, an ion is going to be a charged atom or element. How do we know whether it's going to be charged or not? Well, you need to have a look and see which atom is closest to its nearest noble gas. For example, these are noble gases right here. If we've got something like sodium, right? Sodium is going to be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. It's number 11 on the periodic table. Wants to be closest to neon, which is number 10. So it wants to lose an electron because that has 11 electrons. That is 10. It loses one to be more like neon, and then sodium is left with a charge. So these are the ions. Okay. Now you don't need to do this calculation in the exam, but you do need to understand that an ion is a charged atom or element. It can be positively charged, can be negatively charged. All right. So that's define the difference between the terms atom, element, and ion. What's going to be the next one? I'm just going to double check on the YouTube page to see if everyone can still hear me. Will this be saved for later? Yes, it will. I will be saving this for everybody to watch later on. All right. The next one is outline the 4 major types of macromolecules and how they can contribute to body physiology. So firstly, you need to know that we've got proteins, carbs, fats, which we term lipids, and nucleotides. They're the 4 macronutrients. Now, these 4 macronutrients, proteins, they're building blocks. Carbs, energy. Lipids, also energy, but also storage of energy and protects organs and certain structures, anchors structures as well. And nucleotides are involved in DNA synthesis and also protein synthesis as well. Proteins, so these are the macromolecules. So they're going to be made up of smaller monomers. So these are the polymers, that's multiple subunits. What are the monomers associated with it? Well, proteins are made up of amino acids. Carbohydrates can be made up of predominantly glucose. Lipids are made up of, got to get the whiteboard. Lipids are made up of predominantly triglycerides, and the triglycerides are fatty acids and glycerol. Okay? And nucleotides are made up of nucleic acids. This is A's, T's, G's, and C's. All right? So these are the monomers. The triglycerides is a polymer made up of fatty acids and glycerol. So when you have a look, what are the monomers for proteins? Amino acids. What are the monomers for carbs? Glucose. Could also be fructose and galactose, for example. What are the monomers for lipids? Fatty acids and glycerol. What are the monomers for nucleotides? They're going to be A's, T's, G's, and C's, adenine, thymine, guanine, and cytosine. All right, what's another one? Outline. The physiological significance of electrolytes and how they contribute to pH and the nature and function of buffers. A lot of people get the buffer stuff wrong. Let's have a quick look at the buffers. Firstly, you need to know the definition of a buffer. Buffers resist drastic changes in pH. That's their definition. And when we look at the role of electrolytes in pH, pH is simply the measurement of or concentration of hydrogen ions. pH is equal to the concentration of hydrogen ions. That's the symbol for concentration. So if the concentration of hydrogen ions go up, the pH number goes down because it is an inverse relationship. Don't stress too much about that. But if the hydrogen ion concentration goes up, pH goes down and becomes more acidic. So high hydrogen ions equal acid. Low hydrogen ions equal base. But more specifically, you can define an acid and a base like this. An acid donates hydrogen ions and a base accepts hydrogen ions. That's the definition. All right, that's the first thing. Which means if I were to, so when we look at buffers, buffers are going to be made up of things that donate hydrogen ions and accept hydrogen ions because they need to resist drastic changes in pH. If the pH goes too low, there's too many hydrogen ions, we need to buffer it out. We need to accept those hydrogen ions. If the pH is too high, it's too basic, we need to donate hydrogen ions. So buffers will be made up of acids and bases. Let's have a look. One important one that you need to know for the exam is that bicarbonate buffering system. So CO2 plus H2O gives you H2CO3, which gives you H plus HCO3 negative, right? That's the bicarbonate buffering system equation. Now, what's it mean? It means this. When you hold your breath, carbon dioxide levels increase. Carbon dioxide will jump into the blood from the airways. In the blood, it's made up of predominantly water, binds with water and produces carbonic acid. It's called carbonic acid because we know the definition of an acid. It donates hydrogen ions. There's the hydrogen ion it's donated, and what we're left with is bicarbonate ion. All right. Now, bicarbonate ion is the base here because if it wants to, it can bind to the hydrogen ion and mop it up, absorb it. It accepts it. Therefore, it's a base. That's the acid, carbonic acid. That's the base, bicarbonate. Now, that means that when CO2 levels go up, it pushes the equation in this direction. Hydrogen ions go up. So the more CO2 in the body, the more hydrogen ions in the body, the more acidic we become. That means, now how do we control CO2? By breathing. So if I breathe, hyperventilate, I'm breathing off all that CO2, which effectively means the CO2 decreases, and that means the amount of hydrogen ions in the system decrease. So what that means is this part of the equation here represents breathing, and it tells you that in the short term, we can alter our blood pH by breathing. All right. Now, in the long term, if this isn't sufficient, we can play around with this end of the equation, and the kidneys play around with this end of the equation, and this is long term. Breathing is short term to change pH. the kidneys are long-term to change pH. We can excrete or reabsorb either of those two at our kidneys to alter the pH. All right, make sure you know that for the exam. All right, what's another point we can go through? Discuss the processes of diffusion and osmosis. All right, let's go back to the YouTube page quickly, see how we're all going. All right. So when we want to have a look at the processes of diffusion and osmosis, firstly, let's go through diffusion. Actually, let's just break it up into active and passive transport, because that way we cover a couple of learning outcomes. First one is this: passive versus active transport. We're talking about how do things move across a cell. If I've got a cell here or a cell here, we know that it's surrounded by a phospholipid membrane. Phospholipid membranes do not let anything through that is large or charged. That's the first thing. Doesn't let anything through that's large or charged. Keep that in mind. Now, passive transport. Passive means no energy, doesn't utilize ATP. Active means it does utilize ATP. That's the first difference. Next difference is this: passive is always down the concentration gradient. Active is always against or up the concentration gradient. You know those two things, you're going to be fine. But you also need to know some subcategories of passive and active. For example, if it doesn't require energy and it's going down the gradient, you think of that slide that I talk about, right? If you want to go down a slide, you're up the top, right up here, and you just want to go down the slide, all you need to do is let go and down you go. You slide, no energy. If you want to go back up to the top, you have to climb the ladder. That costs ATP. Low to high, ATP. High to low, no ATP. So another thing you need to know is sitting outside the cell, predominantly we have sodium ions. We've got a whole bunch of sodium sitting outside the cell. We've also got a whole bunch of potassium sitting inside the cell. All right, now, everything wants to go down its concentration gradient. That means if sodium wants to go from outside to inside, it wants to do this naturally, goes down its concentration gradient from high to low. That means it's passive transport. But is it large? No, it's the size of an atom, so it's tiny. Is it charged? Yes, it is. So it can't just freely move through. Therefore, if sodium wants to diffuse into the cell, it needs a channel. Therefore, this type of movement is called facilitated diffusion or channel-mediated diffusion. It's still passive transport. What about potassium? If it wants to get out, it's the same thing. It needs a channel for potassium to diffuse out. It needs a channel, and it's called facilitated diffusion or channel-mediated diffusion. But if potassium, there was a couple of potassium ions outside and it wanted to go in, it's going against its concentration gradient. It's going uphill. Therefore, it requires ATP, and that's called active transport. All right. So there's a couple of different types. There's primary active transport. That's where if I've got all the potassium inside and I've got some potassium outside, if I want to get that potassium from outside to inside, I need a pump which takes that potassium and chucks it in and does it using ATP. That's called primary active transport. Secondary active transport was if potassium, for example, piggybacked on sodium that normally just diffuses in. So if there's a channel here for sodium to diffuse in, if potassium decided to, I'm just going to piggyback on this and diffuse in as well, that's called secondary active transport. That can happen in the same direction, called symport, happen in the opposite direction, antiport. All right. What else we're going to go through? So that's second lecture done. What about the third lecture? Cell, DNA, and hereditary. So if we go through the cell, DNA, and hereditary, what we need to do is go through, identify organelles in the cell and briefly state their function. So the organelles of the cell, we've gone through all this before. Inside the cell, you're going to have multiple organelles. They're small subcellular structures. And what they do is this. You've got the nucleus, houses the DNA. We've got the rough endoplasmic reticulum, has ribosomes. It synthesizes that RNA from the nucleus and synthesizes proteins from that RNA. These proteins are destined for exportation. That's what the ribosomes sitting on the rough endoplasmic reticulum do. It also then transports those proteins to the Golgi apparatus, which then modifies and packages those proteins for exportation out of the cell. We've also got the smooth endoplasmic reticulum, which is important for detoxification, therefore high levels in the liver, and also for lipid production as well. You've got peroxisomes, you've got lysozymes, you've got all these different other subcellular structures. Make sure you go through them and you know what they do. Understand the basics of Mendelian inheritance of somatic and sex chromosomes. All right. A couple of people got these wrong in the mid-trimester exam. It's easy. Let's go through a couple of important points. First important point when it comes to inheritance is this. You've got 23 pairs of chromosomes. Of these 23 pairs of chromosomes, 1 to 22, they're the somatic chromosomes, and 23, that's the sex chromosomes, right? All right, now what does that mean? It means this, they're inherited differently. First thing is this. For the somatic, 1 to 22, if I said to you that my parents, both of them, had a recessive gene in chromosome 1, that means, for example, recessive inheritance is where you require 2 copies to get the particular phenotype. That's the way you present, okay? So if it's chromosome 1, if a big copy of chromosome 1 meant healthy and a little copy meant mutated, then for mum's going to have a pair of chromosome 1. I've got a pair of chromosome 1. Dad's got a pair of chromosome 1. She only gives me 1. Dad only gives me 1. Therefore, I've got my pair. Same happened for my mum and my dad. So if mum is a carrier for a recessive disease that sits in chromosome 1, then she's going to have a big one and a little one. All right. Now, if she had the disease in chromosome 1 that was recessive, she'd have a little one and a little one. If she did not have the disease and was not a carrier, she'd have big one, big one. Okay, hopefully that makes sense. Because when it comes to inheritance, when we're looking at Punnett squares for somatic chromosomes, we've got mum, we've got dad. Let's say they're both carriers for a recessive disease that sits in chromosome 1. That means mum's going to have big one, little one. Dad's going to have big one, little one. And then you can see how they have potential offspring. What happens is big one can be with big one, little one, big one, little one, little one, little one. And what we end up having is this. 25%, because there's 4 here, right? 4 possible outcomes. 25% of the offspring, normal, uneven carriers. 50% of the offspring are carriers. 25% of the offspring will have the disease, all right? This is how it happens for chromosome 1 all the way up to 22. But if I ask you a question in the exam about chromosome 23, which is the sex chromosomes, well, then something else happens. Sex chromosomes are this. Dad, right? Actually, I had mum there before, so let's have mum. Mum is XX. That's her sex chromosomes. Dad is XY. And what are the potential outcomes for offspring? XX, XX, XY, XY. That's why mum and dad could potentially have 50% chance of having girls, 50% chance of having genetic boys, all right? Now, what about diseases? Well, this is an important point. If mum is a carrier for a disease in the X chromosome, so she needs 2 copies. She's only got a mutation in one of them. What happens is this, XX, normal, XX. This one's got the mutation, XY, normal, XY, mutation. All right, what does this now mean? It means that 50% of the girls will be carriers if mum was a carrier. It means that 50% of the boys will have the disease because they don't have a second chromosome X to make up for that damaged one. They're going to get the disease, okay? That's what happens with sex chromosomes, all right? All right, now what are we going to go through? So that was the chromosomes. Let's have a look at, based on the plasma membrane structures. Oh, I went through active transport, passive transport, diffusion. I didn't go through osmosis and tonicity. Let's go through osmosis and tonicity. All right, so if, again, I have a cell and there's a concentration difference from one side of that cell to the other, let's say for sodium. What happens is this. If you've got a lot of sodium outside, but not much sodium inside, okay, and you want to balance it out, because remember, the body just wants to balance out concentrations from one side of a membrane to another, just wants to balance, 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 balance. If it's not balanced and the ions or the solutes can't move, then the solvent moves. What's the solvent? Biologically, it's always going to be water. So what happens here? In this case, if it's more concentrated outside the cell than inside the cell, the water will move out. Okay, this is osmosis. If it's more concentrated inside, the water will move in. Therefore, you can define it as though osmosis is the movement of water from an area of high water concentration to an area of low water concentration. You may be thinking, why is it low water out here and high water out here? Because there's more solutes out here taking up the space. Less solutes here taking up space, therefore more water here, less water here. If this happens, now think about this clinically. All right? If you're studying to be a nurse or a doctor, what's going to happen here? Normally, the concentration of this stuff is balanced from outside to inside, right? Therefore, if you inject something into a patient and it's a high concentration of something, and it's going to sit outside the cell because it's gone from your bloodstream to the tissues compared to inside, that means you've now just injected a hypertonic solution. This is hypertonic. Hyper means more than. Tonic is referring to concentration. That means water wants to balance it out. If these can't move through the cell membrane, which they likely can't, water will move and water rushes out. If water rushes out, once you've given somebody a hypertonic solution, the cell shrinks, and that shrinking is called crenation. All right, so remember that. The opposite. If hypotonic solution is represented outside the cell, that means it's less concentrated outside the cell compared to inside. Water will move in the opposite direction, and water will move in to balance out the concentration, and the cell will swell. Okay, they'll swell so much that it will lyse, and if it lyses, that means it bursts. Right, so that's tonicity. All right, let's check the YouTube page. I'm in the Facebook page, see everyone's going. All right, so what now is we're going to have a look at tissues and the integumentary system. All right, so what do you need to know about the tissues? Well, know that when we look at tissues, you're going to have different types of tissues. Connective tissue, muscle tissue, nervous tissue, and epithelial tissue. What do you need to know about each? So remember that connective tissue is made up of cells, gels, and fibers. All right? The cells depend on the type of connective tissue, because connective tissue can be bone, connective tissue can be blood, connective tissue can be cartilage. They've all got different cell types associated with it. The gels are going to be everything in between, right? Like the cement mix. And the fibers are going to be embedded in, and it determines the property of that particular type of connective tissue. If it's, remember, the three different types of fibers in connective tissue are elastic, reticular, and collagen. If you want tough tissue, it's collagen. Stretchy tissue, elastic. And if you want tissue that's going to play some sort of filtration role, or maybe it's going to be a type of tissue that needs to be distendable or be able to be squished and moved, reticular. Right? Muscle tissue, three types: smooth, cardiac, and skeletal. Smooth muscle, cardiac muscle, unconsciously controlled, okay? Skeletal muscle, consciously controlled. It's called skeletal because it's attached to your skeleton. So biceps, tongue, face, legs, they're all going to be skeletal muscle. Cardiac muscle is the heart. That's an easy one. Smooth muscle, it lines hollow organs, digestive system, including esophagus, small intestine, large intestines. It can also be that of the urinary system. It could also be that of the reproductive system. All right. Nervous tissue, what do you need to know? You need to know about neurons and glia. Neurons send the electrical signals. Glia, they support the neurons in their role. What about epithelia? What do you need to know? Epithelia, remember, form equals function. Remember that. We'll be going through epithelia in 1 second. So form equals function. And when we look at form equals function, you can have different types. You can have simple epithelia, that means one layer. You can have stratified epithelia, that means more than one layer. You can have squamous epithelia. That means squished. You can have cuboidal. That means it's shaped like a cube. You can have columnar. That means it's shaped like a column. Therefore, you can have simple columnar, one layer of column-shaped cells. Stratified squamous, many layers of squished cells, okay? And the form equals the function. If it's stratified squamous, it's many layers and squished. It's there for protection, all right? Okay, what do you need to know when it comes to the integumentary system? All right. When it comes to the integumentary system, make sure you just go through basically the general layers. So epidermis, dermis, hypodermis. Now there's multiple layers in the epidermis. You don't need to know them. Dermis, hypodermis. Epidermis has those multiple layers. The cells are sitting in the epidermis. So the keratinocytes, melanocytes, Merkel cells, Langerhans. All these cell types sit within the epidermis, okay? You got Merkel, touch receptors, Langerhans, they're there for immune system function. Keratinocytes are there for protection, and melanocytes are there for UV protection of the cells. The dermis, this is where those bodies of the hair follicles are going to be sitting. This is going to be where all the sweat glands and oil glands sit as well. And the hypodermis is basically all just fat. All right, so just know those major differences, you'll be fine. All right, skeletal system. Explain the function of each of the three types of cartilage. So 3 types of cartilage include, you're going to have fibrocartilage, elastic cartilage, and you're going to have the hyaline cartilage. Nearly forgot. All right, fibrocartilage, what's important about that? Fibrocartilage resists compressive forces, and it's found in the spine, right? Elastic cartilage, it's going to be distendable, and it's found in the ear and epiglottis. You get the hyaline cartilage, and it lines ends of long bones. So for example, if I've got a bone like this, which looks like that off the Flintstones, lining the end of that long bone is going to be hyaline cartilage because it's there to reduce friction. All right. Describe the basic structure and function of bone and how different cell types contribute to bone physiology. All right. For example, if we look at long bones is going to be a bone that looks like that, for example. All right, and we've got the diaphysis. We've got the metaphysis. Sorry, the metaphysis. And the epiphysis. And you've got the distal. So proximal, distal, right? Now, you've got a hollow inside. It's called the medullary cavity. You're going to have compact bone, and you're going to have spongy bone. Inside the medullary cavity of long bones is going to be yellow bone marrow. For adults, in a child, this medullary cavity has red bone marrow. That's the site for red blood cell production. The different cells. Okay, remember the different cells of bones include the osteo. So osteo is the prefix for bone. And then blast means to build. They build the bone. You got osteoclasts. They crush the bone. You got osteoprogenitor cells. They're stem cells. Then you got osteocytes. They're mature osteoblasts. So just remember, the blasts build, the clasts crush, all right? The sites, they stay there and they just determine what's going on. Does it need to build? Does it need to crush? Whatever. Outline the process of calcium homeostasis. All right. All right, calcium homeostasis. Let's have a look. First thing is, you've got your thyroid gland. Behind the thyroid gland is the parathyroid gland. It releases parathyroid hormone. So here's the thyroid gland. Here are the parathyroid glands sitting on the back of the thyroid. When blood calcium levels are low, it stimulates the parathyroid gland to release parathyroid hormone. Parathyroid hormone with vitamin D, which is coming from sunlight and our skin. Do a couple of things together. Together, what they do is they increase calcium absorption at the intestines, at the small intestines, right? They decrease calcium excretion at the kidneys, and they increase osteoclasts to break the bone down. All of these have the desired effect of increasing blood calcium levels. See how that's negative feedback. Stimulus was low blood calcium. The receptor was the parathyroid gland, but also it was the control center, released parathyroid hormone, which together with vitamin D, increased calcium absorption at the small intestines, decreased calcium excretion at the kidneys, and increased osteoclast activity at the bone, breaking the bone down, releasing the calcium into the blood. List the major bones. You can do that yourself. There's a list there. Understand the basics of synovial joints. Just make sure you go through with the synovial joints, the 6 different types, and whether they are multiaxial, biaxial, uniaxial, and so forth. All right, lecture 6, fundamentals of the nervous system. We're getting into the fun stuff. So you've all done an exam on everything up until this point. This is the stuff you haven't done an exam on. So, compare and contrast the structural functional divisions of the central nervous system, peripheral nervous system. So, this one's easy. When you've got the brain, that's a terrible brain, but you've got the brain, brainstem, spinal cord. This is the central nervous system. Brain, brainstem, spinal cord. Then you're going to have all the nerves that shoot out and away and come back in. That's the peripheral nervous system. They're the 2 major divisions, all right? With the peripheral nervous system, you can break it up into sensory and motor. All right, these are things coming in, these are things going out. What's coming in? What type of sensory input is there? Well, it's going to be coming from our skin and outer body. This is somatic. And coming from our organs called visceral. All right, so somatic from the body, visceral from inside the body. For motor, you're going to have basically somatic, which is all conscious movement. Then you're going to have autonomic, which is unconscious movement. And this then gets broken up into the sympathetic nervous system and parasympathetic nervous system. All right, list the cell types, neurons, glia, and describe their anatomical physiological significance. All right, a couple of glia that you need to know. First, glia, they're supporting cells. What do? You got ependymal cells. They make the cerebral spinal fluid in the ventricles. You've got the microglia. They're actually immune cells. You've got oligodendrocytes. They make the myelin sheath that wraps the axons in the central nervous system. You've got the Schwann cells. They do the exact same thing, but in the peripheral nervous system. Okay, ependymal, microglia, oligodendrocytes, Schwann cells. What else are there? It's a couple of different types. I can't remember all of them right now, but make sure you go back through them. Remember, if you have any questions, just pop them up. If there's anything you want me to specifically go through, just ask. All right, explain. And for the neuron, if you've got a neuron, right, that's how I draw my neuron. You got the soma, which is the body. You're going to have a nucleus as well. You got the axon, which is going to be surrounded by myelin. Then you got the axon terminals as well. And on the soma, usually you've got dendrites, and they usually receive the signal coming from the previous neuron or from wherever it's coming from. All right, explain the physiological basis of the generation and transmission of a nerve action potential, including the role of the important ions and channels. This is a big one. All right. Yes, we'll go through muscle contraction. That's coming up. So if we first go through the action potential, you need to know the action potential. If you can do this, you'll be fine. If you can draw up a graph, and on that graph you can put minus 70, minus 55, 0, positive 30. So if you can do that, right, that's going to be voltage, so that's millivolts. Down here is going to be time in milliseconds, right? Now we know that a neuron, the resting membrane potential is going to be sitting in around about -70 until it starts to get stimulated. Now when it stimulates, channels open up. If these channels open up enough, let's draw it up over here. Right, actually, let's put a neuron here. Right, at rest, we know it's negatively charged inside the cell compared to outside the cell where it's positively charged. I put an electrode in. The difference between outside and inside, -70. Now what happens is I slowly stimulate this neuron. It opens up some channels that are specific for sodium. Some sodium leaks into the cell. Takes its positive charge with it, and it makes this part of the neuron slightly positive. It starts to bump up. If enough positive sodium go in that it hits the threshold, -55, then all the voltage-gated sodium channels along the axon open up in a domino-like fashion. And all this positive sodium rushes in, carrying its charge with it. That's why it spikes up to positive 30. Then, at positive 30, the sodium channels shut. At positive 30, potassium channels open up. And potassium's inside the cell. Therefore, if their channels open up, potassium leaks out, taking its positive charge with it. Therefore, this drops back down. So much potassium leaks out that it goes below -55, goes below the resting membrane potential -70, goes down to around about -90. And then everything's the opposite. All the sodium's in, all the potassium's out. That's the opposite of a normal resting cell. How do we swap it back over? That's when we get the sodium-potassium ATPase pumps. Throws potassium in, throws sodium out, then it resets it, and now we're back to normal. That's an action potential. You need to know. That's the resting membrane potential. This part's called depolarization. That's where sodium comes in. This part's called repolarization. That's where potassium goes out. And this is called hyperpolarization. And that's where more potassium goes out. All right? So that's an action potential. You need to know that. All right. Describe a synapse. Outline events associated with synaptic transmission. So we just spoke about the action potential going down a neuron. What happens when it gets to the end and it wants to speak to the next neuron? That's called synaptic transmission. When we have a look at synaptic transmission, what happens is this. And here's the next neuron. So we've had our action potential. All this sodium's coming in. Boom, 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 boom. So all this positive sodium's coming in. Now we hit the end of the neuron, right? The axon terminals. All this positive stuff coming in triggers a new type of voltage-gated channel, not a voltage-gated sodium channel, but a voltage-gated calcium channel. And the lid pops open, and calcium jumps in to the axon terminal of the cell, of the neuron. This then triggers the release of these small little vesicles that contain neurotransmitters, tells them to then bind to the axon terminals. They then bud off and release their contents, and they diffuse across the synapse and bind to their receptors on the other side. When they bind to their receptors, that leads to the opening of more sodium channels. And now sodium diffuses in, and now the action potential is sent down the next neuron. All right, so that's synaptic transmission. All right, describe the basis neurotransmitters. Okay, when we go through the neurotransmitters, you don't need to know a lot of details about those particular neurotransmitters. What you need to know is, I'll show you in a second. Just checking the YouTube page, the Facebook page. All right, so when we look at the neurotransmitters, all you need to know really is this. Now write the full word. Don't forget that. Acetylcholine is abbreviated like that, but you need to write the full word to get full marks. Acetylcholine is involved in skeletal muscle contraction. Acetylcholine is involved in memory in the central nervous system. Acetylcholine is involved in the autonomic nervous system. Right? When you look at adrenaline or noradrenaline, it's involved with the sympathetic nervous system. When we have a look at dopamine, reward, and movement in the basal ganglia. When we look at substance P, pain transmission. And don't worry about serotonin, GABA, and glutamate. Glutamate, excitatory neurotransmitter. GABA, inhibitory neurotransmitter. All right, now let's look at central nervous system. So go through identify the following areas of the brain and their function. All right. Some important points when we go through the central nervous system with the brain and their function. So you're going to have, firstly, divisions. Now, these divisions include the frontal lobe. Now, I'm talking about the cortex here. Cortex is the outer couple of millimeters of the brain itself. Frontal lobe, parietal lobe, occipital lobe, and temporal lobe. You've also got the insula, which if I move that temporal lobe away, you're going to have a basement part of the brain. That's going to be the insula. That's therefore gustatory sensation, so taste. All right. What's important here is this. Occipital, there for vision. Temporal, there for hearing. Frontal and parietal. Now, other things, but that's all I want you to know at the moment. But frontal and parietal is this. Parietal lobe is for sensory. Picks up and understands sensory information. Frontal, motor information. It begins there. All motor is going down and away, right? All sensory is going up. Therefore, sensory makes sense here. Motor begins here and goes down to the body. So remember that part. When we look at the diencephalon, thalamus, hypothalamus, epithalamus, a couple of things. It's going to be sitting around about here. You're going to have the hypothalamus with the pituitary glands underneath. The thalamus is going to be sitting deep in the brain around about there. The thalamus is the sorting center, right? It's like the post office. Takes information, determines where it needs to go. Hypothalamus is the master regulator of the sympathetic nervous system and the endocrine system as well. The epithalamus is involved with the pineal gland, has important roles in sleep-wake cycles. What about the brainstem, midbrain, pons, and medulla? All you need to know about that is that it's very important when it comes to breathing, so respiration, cardiovascular function, basic functions as well, reflexive, like cough reflex and so forth. And the cerebellum, which is that little brain that sits at the back, right? The cerebellum is very important when it comes to balance, coordination, proprioception, right? So balance, coordination, proprioception, so very important in modulating your movement. Let's have a look at describe the structure, function, and protective mechanisms of the central nervous system, including blood-brain barrier, meningeal layers, and cerebral spinal fluid. Okay. Blood-brain barrier is a mechanism that selectively chooses what it wants to go from the blood to the brain. And there's a couple of different layers of the blood-brain barrier. It includes the endothelium, so that's the blood itself. So the endothelia have these tight junctions that don't let anything through. So the endothelial layer is one mechanism. You're also going to have a basement membrane underneath that. You're also going to have these pericytes, which are glia that actually have little legs that sit on top. Right? And so they're glia. And these things all together play an important role in selecting what comes through the bloodstream and goes to the brain and what doesn't get to the brain. The meningeal layers are a little bit different. The meningeal layers, you can actually, because you can't see the blood-brain barrier, but you can see the meningeal layers. If you've got the brain itself, you're going to have the meningeal layer that's closestly adherent to the brain, which you could draw like this, which is called the pia mater, which means soft mother. Then you're going to have the next layer, which is called the arachnoid mater. It's called that because it's got these spider-like projections. It's the arachnoid mater, right? And then the last one is the dura mater, which means tough mother. It's like a paper bag that sits on the outside. Right? Now, the arachnoid mater, in this subarachnoid space, that's where our cerebral spinal fluid floats around. And then that last thing leads us into cerebral spinal fluid that is produced. Cerebral spinal fluid is produced by the ventricles of the brain, right? And if we were to look at the ventricles of the brain, you're going to have two lateral ventricles. Let's just draw it up a little bit better. You've got four ventricles, basically, of the brain. These are these hollowed pockets within the brain. They're hollowed out from when we began our journey to create the nervous system as a hollow tube, and the foldings and undulations resulted in these two right? And so what you've got is a paired ventricles here called the lateral ventricles. Then you've got the third ventricle, and you've got the fourth ventricle. Where does it sit? It's basically going to be sitting like this. I'll draw the brain over the top of it, like that. So that means the lateral ventricles are sitting deep within the cerebellum itself. So the third ventricle is sitting near the thalamus and hypothalamus, and you can see the fourth ventricle is sitting right where the pons is, and behind it's going to go close to where the cerebellum is. And again, ependymal cells that sit within these ventricles produce cerebral spinal fluid. Okay, let's look at the sensory and motor tracts. A lot of people have difficulties with the sensory and motor tracts. Let's have a look. I'm losing my voice a little bit, so I'm sorry about that. How long have we been going for? 54 minutes. God, no wonder. All right. So if we have a look at sensory tracts, firstly, let's draw up the brain. All right. Let's label some stuff. Let's put the thalamus in here. We've got the midbrain, the pons, and the medulla. Midbrain, pons, medulla. All right. And so we're talking about sensory now. Let's look at sensory signals. Fine touch. Somebody touches my hand finely. It gets a pen, maybe a feather, tickles my hand. A bit of sensory information is going to be coming in, right? Once it gets into my spinal cord, it goes straight up to the medulla. That's where, now remember, this is fine touch. What's the other name for fine touch? It's the dorsal column medial lemniscus pathway. Good name. Dorsal column medial lemniscus pathway. You need to know this. Fine touch goes into the spinal cord, goes up the same side to the medulla, where it synapses with the next neuron, where it crosses over, called decussation at the medulla. Then it goes up to the thalamus where it synapses with the third neuron, and then goes to part of the parietal cortex involved in somatic sensation of the hand. It goes, you've now touched your hand. How many neurons here? 1, 2, 3. Fine touch. What about pain? What if I had a painful stimuli that was coming from my hand as well? Well, it goes in. So this is now pain or temp. Pain or temperature, right? Goes in, synapses once it goes in with the second order neuron and crosses to the other side, decussation, crossing the other side, then goes up. Doesn't synapse at the medulla like fine touch does, but does at the thalamus, because remember the thalamus is the relay center. Synapses, goes to a part of the brain that deals with the hand and says, hey, it's pain. What's the difference between pain, also known as the spinothalamic pathway? What's the difference between pain and fine touch? Where it synapses, where it crosses over. All right, make sure you know that. What about motor? Well, let's just do motor here. Motor is going to come from the frontal cortex, right? The prefrontal cortex. It's going to send a signal that goes down, and basically motor will go down and it will synapse pretty much at the level in which it needs to leave the body, okay? I've actually buggered that up because it's going to cross over at the medulla. It goes down, it's going to cross over at the medulla, and then it's going to synapse at the level. So you get the upper motor neuron and the lower motor neuron, all right? Okay. We're getting there. All right, lecture 8, peripheral nervous system. Outline the sensory processing, reflexes, pain, temperature, touch, chemical stimuli. All right, something important here is this. When you look at the spinal cord, and we're to draw the spinal cord up, right? Cross section. You've got the grey matter here and the white matter here. You're also going to have the spinal nerves coming out on both sides. All right, need to be able to label this. All right, so you need to know grey matter, dorsal grey horn, ventral grey horn. So that means this is dorsal, that's back. This is ventral, that's belly, that's front. Right? You got the, what's this? This is the ganglia. This is the dorsal root ganglia. All right, and it goes to the mixed spinal nerve. All sensory information will come in via the back, always, always, always. That's where the ganglia sits. That's the body that sits outside the central nervous system. Right? All motor information will leave via the front, always, always, always. Don't forget that, okay? All right. Let's have a look at 12 cranial nerves. I'm not going to do a lot of detail here because that could be a whole hour in itself. Just remember the tip I gave you when it came to remembering them, all right? Now, you do need to know for the exam, you need to know the names of the cranial nerves, the number of the cranial nerves, whether it's sensory or motor, and also its function. So remember, O, 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 to touch and feel very good velvet heaven, right? O, 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 to touch and feel very good velvet heaven. So first letter of each, optic, bugger it up. So olfactory, optic, oculomotor, trochlear, trigeminal, abducens, facial, vestibulocochlear, glossopharyngeal, vagus, accessory, and hypoglossal. And then you say, some say, marry. So that's just going to be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So remember, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Question is, Will you have the numbers for the cranial nerves in Roman numerals, for example? Maybe I will. I can't remember what I wrote down. I'm pretty sure I just wrote the name or could have the number, the Roman numeral number. So make sure you remember it. Okay. Remember, some say marry, money, but my brother says big brains matter. What have I missed? Oh God. No, I buggered it up. Some say marry, money, but my brother says big brains matter most. S, sensory. M for motor. B for both. All right, so remember that. Remember their function. All right, olfactory, sense of smell. Optic, vision. Ocular motor, moving the eye. Trochlear, moving the eye. Trochlear means pulley. Trigeminal is going to be both sensory and motor. Sensory for face, motive for chewing. Abducens is going to be to abduct the eye away. Facial is going to be moving the face, but also anterior taste of the 2/3 of the tongue. Vestibular cochlear is going to be balance and sound. Glossopharyngeal is going to be swallowing, but it's both. So glossopharyngeal is also sensation at the back of the tongue and swallowing as well. Vagus is going to be everything below head and neck, so heart, lungs, so forth. Accessory is going to be shrugging the shoulder, and hypoglossal is moving of the tongue. All right, there's your 12 cranial nerves. Compare and contrast. Oh, we're going to do the plexuses. What do you need to know about the spinal nerve plexuses? All right. So what you need to know about the spinal nerve plexus is this. I'm just going to check the Facebook page again. See if there's any questions on there. Okay, so when we look at the spinal nerve plexuses, what do you need to know? You need to have a look at the, firstly, cervical plexus, brachial plexus. You need to look at, remember, there's no thoracic plexus, but there's a lumbar plexus. There is a sacral plexus. All right. And when we look at these 4 plexuses, so you're going to have cervical plexus. What do you need to know? Well, you need to know that the cervical plexus does the back of the head, the neck, and the shoulders. And of the cervical plexus, a little bit of the brachial too. You have C3, C4, C5, C3, C4, C5. So C3, C4, C5 keeps you alive, is what we say, and that's the phrenic nerve, and the phrenic nerve innervates the diaphragm. Remember that. Now, with the brachial plexus is going to have nerves so it's going to be around about C6 to T1, and it's going to have nerves that do the radial nerve, ulnar nerve, and median nerve. All right. Radial moves the thumb, median going to be the middle, ulnar is going to be moves its way around the elbow and pinky finger as well. Now, with the lumbar plexus, you're going to know that you're going to have the femoral nerve. And what the femoral nerve does is it basically anterior, medial, and lateral aspects of the thigh. And then when we look at sacral, you're going to have the sciatic nerve. Basically does the arse, back of the thigh, and everything below the knee. So they're your cervical plexuses. All right. Okay, now what do we need to know? We need to go through, compare and contrast sympathetic, parasympathetic nervous systems. What do you need to know for that? All right. Both the sympathetic and parasympathetic nervous systems, which I can draw a little bit differently because they are different lengths. All right. When we look at the sympathetic, parasympathetic nervous systems, first one is this is the sympathetic. That's fight or flight. This is parasympathetic. That's rest and digest. Both are 2 neuron chains, are part of the autonomic nervous system. You get scared, sympathetic nervous system. What happens is when you're resting, digesting, parasympathetic nervous system. So let's just say both of them want to ultimately innervate the heart, right? Sympathetic nervous system is going to increase its contractile force and speed, right? Parasympathetic nervous system is going to decrease the contractile force and decrease the speed, right? How does it do it? Like this. Preganglionic neuron is the first one. Preganglionic, that's for both. Postganglionic, for both. Don't forget those terms. For both the sympathetic parasympathetic nervous system, they both release acetylcholine. That's the neurotransmitter. And they both bind to nicotinic receptors at the postganglionic neuron. Now, the difference is at the postganglionic neuron, what neurotransmitters do they release? Sympathetic nervous system releases noradrenaline. Parasympathetic nervous system releases acetylcholine again. All right. And the receptors for the parasympathetic nervous system on the organ is going to be muscarinic. And for the sympathetic nervous system, it's going to be adrenergic. So know that, right? Now, the sympathetic nervous system, if it'll take, I'll rub this out, where does it exit the body, right? So where do these neurons exit the body for the sympathetic versus parasympathetic? It's easy, but don't forget it. I draw the brain up. For the sympathetic nervous system, all those first preganglionic neurons come out of the thoracic or lumbar region, right? Which we term thoracolumbar. That's the sympathetic nervous system. The parasympathetic nervous system, all the neurons, preganglionic neurons, come out of the cranial region or the sacral region, and that's termed craniosacral. Parasympathetic nervous system. All right. What else do we need to know? Okay, physiological stimulation of the receptors. You do need to know this. When you look at the sympathetic nervous system, fight or flight, and we look at the adrenergic receptors, there's a couple of different types you need to know. You've got alpha 1, beta 1, alpha 2, beta 2. Now there's a beta 3, don't worry about it, it's found only in fat cells. Section them off like this. Then right, if you hit these with adrenaline, stimulates them. If you hit these with adrenaline, it inhibits. Now where are they located? And we can determine what they do. So alpha 1. So let's start beta 1, beta 2. Beta 1 is found in the heart. Beta 2 is found in the lungs or airways, right? Alpha 1 is going to be found on all smooth muscle and glands, right? Alpha 2 is presynaptic membrane. Firstly, don't stress about that one for the exam, but know these ones. So that means if I throw adrenaline at alpha 1, it's stimulating all my smooth muscle. Therefore, the smooth muscle on my peripheral blood vessels, vasculature, are going to constrict, shunting the blood inwards. That's important for fight or flight because it wants to move that blood to areas like my skeletal muscles where the blood vessels will dilate. That's going to be in control of actually that one, to be honest. And then all the blood goes in. If you throw adrenaline at beta 1, the heart's going to contract. It's going to increase its contractile force, increase its speed. That's what happens when you throw adrenaline at beta 1 because it's stimulatory. If you throw adrenaline at beta 2, it's inhibitory. It relaxes the airways. They open up, more air can come in and out. Don't forget that. All right, we're getting there. Endocrine system. All right, done with the nervous system. Explain the different types of hormones, controls of secretion, mechanism of action, and receptors. Explain the structural. Okay, so first thing is this. Different types of receptors and different types of stimulation. So how can you stimulate hormones to be released from endocrine glands? So you can have neural stimulus, hormonal stimulus, and humoral stimulus. Neural stimulus is when neurons stimulate hormones to be released. Hormonal stimulus is when other hormones stimulate hormones to be released. Humoral stimulus is when products in the blood stimulate hormones to be released. So for example, neural stimulus is when a neuron, for example, activates the adrenal gland, right, releases hormones. Hormonal stimulus is when one hormone goes to the bloodstream, tells another gland to release a hormone. They're usually called the tropics. And humoral is when there's products in the blood, like glucose. Glucose can stimulate hormones to be released from the pancreas. All right, let's look at the pituitary gland and the hypothalamus. The base of the brain, I showed you where the hypothalamus was, and you're going to have two connections there. You're going to have the pituitary gland, one's going to be anterior, one's going to be posterior. And the posterior pituitary gland is directly connected, anterior pituitary gland is not. There is actually nerves that go from the hypothalamus to the posterior pituitary gland, which means the posterior pituitary gland is neurally stimulated to release its hormones. It produces, in actual fact, hypothalamus will produce the hormones that the posterior pituitary gland hold and release. These are ADH and oxytocin. Right? ADH, antidiuretic hormone, tells us to not pee, hold on to the water. Oxytocin stimulates labor contractions and also stimulates milk ejection. Right? When it comes to the anterior pituitary gland, there is a vasculature supply. There's blood vessels that actually come down to stimulate it, which means they produce and secrete their own, but only in response to the hypothalamus, which means the hypothalamus must release a hormone that travels through this, and that's called a hormonal stimulus. What are the hormones that the anterior pituitary gland produce and release? We've got prolactin. Prolactin plays an important role with milk production. We've got adrenocorticotropic hormone, travels to the adrenal gland, tells it to release mineralocorticoids and corticosterone, for example. You've got growth hormone. You've got thyroid stimulating hormone, and you've got the gonadotropins, which are follicle stimulating hormone and luteinizing hormone. All right. Okay, so for the exam, what do you need to know for these? You need to understand what's being produced by each and the function of each as well. Okay, next part that we need to go through. Any other questions? Let's go check the Facebook page. Okay. So, outline the major effects of hormones released from the anterior pituitary. We've done that, thyroid and parathyroid. We've done parathyroid. Thyroid hormone, well, thyroid releases thyroid hormone. That's T3, T4. T3, T4, the number tells you how many iodine molecules are attached, and their major function is that for metabolism. You have more thyroid hormone, increased metabolic activity. That means increased weight loss and also heat production, sweating, so forth. And the opposite happens for low thyroid. Adrenal gland, it releases adrenaline. That's in the medulla. In the cortex, it releases the mineralocorticoids like aldosterone. It plays an important role for holding on to sodium, which holds on to fluid. And in the cortex, you also got the corticosteroids, the steroids like cortisol, for example, which gets released in times of stress, relaxes the immune system, but also what it does is it can stimulate the adrenal cortex and go to the brain and tell a whole bunch of other hormones to be released. And the pancreas, which releases, when blood glucose levels are low, the pancreas will release insulin. When blood glucose levels, other way around, I'm so stupid. When blood glucose levels are high, it releases insulin. Blood glucose levels are low, it releases glucagon. Insulin are the keys to open the door of the cells for glucose to come in. Glucagon tells the liver to start breaking down the glucose glucose that's stored in the form of glycogen. It's released. Perfect. All right, nearly there. How long would be? An hour and 14 minutes. That's way too long. Muscular system. Okay. When we look at the muscular system, what do we need to know? All right, compare and contrast the physiological difference between the three types of muscles. Well, we've already done that. When we look at three types of muscles, you're going to have smooth, which looks like a spindle or an eye. It's got a single nuclei. You're going to have skeletal, which is cylindrical. You can have one or more nuclei. And you're going to have cardiac, which is branched like that, and it's branched because other cardiac cells are attached to it like that, and it can be one or more nuclei. Now the skeletal and cardiac are striated. Smooth is not. It's called smooth because it's not striated. The striations are the filaments that are inside that tells the muscles to contract over the contractile filaments. I'll talk about those in a second. Okay, outline the physiological processes regulating transmission at the neuromuscular junction. All right. Similar to what we spoke about before, you're going to have a neuron, and this time it's going to synapse not with another neuron, but it's going to synapse with skeletal muscle. Now, skeletal muscle has these tubules. These tubules lead to the endoplasmic reticulum equivalent called the sarcoplasmic reticulum, which are little storage units, and they're filled with calcium. All right, and inside you're going to have all these filaments that need to cross over each other. All right, first thing that happens is this. The sodium through the action potential jumping into the neuron, we know this. As it gets to the end, it opens up voltage-gated channels for calcium. We know this. That releases all the vesicles containing the neurotransmitter. In this case, it's acetylcholine. So now acetylcholine is released into this synaptic cleft. This is called the neuromuscular junction. It must bind to acetylcholine receptors. Once it does, it opens channels for sodium again. And more sodium rushes in. This time, sodium rushes in along the membrane. Now, we want to tell the deep fibers of the muscle to contract. But because it's membrane potentials, it's only happening at the membrane. So how do we get deep in? Well, we need to go down these T tubules. So now the sodium diffuses down these T tubules. And once it gets down, hits the sarcoplasmic reticulum filled with calcium. And all the calcium is now released. Sodium, then calcium. Sodium, then calcium. Once calcium is released, it can now tell the skeletal muscle to contract. How does this happen? This is the next part. All right. You are going to have thin and thick filaments within muscle. And what you've got is myosin and actin. Myosin have these golf club looking things. It's got these bodies with these, it's got this, what looks like a club with the head of the club, and they've got binding pockets on the actin. Now the thing is this, the actin is all bound up. There's a chain around it. And this chain, like a bike chain, has a lock on it. This lock needs to be unlocked in order for the chain to move away. Great thing is, the first thing, this lock is called troponin. The chain is called tropomyosin. And once calcium has been released from what I said earlier, calcium comes along. It unlocks tropomyosin, so calcium's the key to unlock the lock. Tropomyosin disappears. The bike chain falls away, and now what we've got is open actin, which now these myosin heads combine to. So calcium is the key. Unlock the troponin for the tropomyosin to fall away. Now binding can happen. How does binding happen? We need ATP. This is basically what happens, right? You've got the actin and the myosin heads. Now, ATP comes along, binds to the myosin head, and splits to ADP and phosphate. That cocks the head into position and binds. Then the ADP, the phosphate disappears and it moves. Now what needs to happen is we need more ATP to release that myosin head. All right, so ATP comes along and binds to the myosin head, it pops off. Then it splits into ADP and phosphate and cocks its head again and binds and then falls away and moves. More ATP is required. So what do we need ATP for? We need ATP to cock the head and binding, contraction, but then we need more ATP to release. This is what happens in rigor mortis. After death, all this calcium is released, so all the tropomyosin and myosin fall away. Then the ATP that's present allows for contraction to occur, but then all the ATP runs out. When ATP runs out, there's no ATP to unlock that head. so the muscles stay contracted. So what happens with ATP? ATP is required to cock the myosin head, bind, and contract. All right, remember that. Calcium unlocks the chains for it to happen, unlocks troponin and tropomyosin. ATP is there for the contraction itself. All right. And then the next one is identify major clinically relevant skeletal muscles. That's up to you. Go through the list. Hematology and immunology. All right, we're nearly there. First thing, most important thing you need to know is with the hematocrit. Hematocrit is going to be the blood. I want to take some of your blood, spin it down. You're going to have these different layers, all right? Now, these different layers are going to be plasma, white blood cells, and platelets. And then you're going to have the red blood cells. Plasma is the majority. Make sure you know the percentages for each. What percentage sits within each? That's important. That's called a hematocrit. Next thing that you need to know is hemostasis. Again, don't get this mixed up with homeostasis, right? You need hemostasis. Now, for hemostasis, what we need is the 3 different phases of hemostasis. First is vascular spasm. Second is platelet plug. Third is clot formation. So remember, you cut yourself. Hemostasis means let's stop the bleeding. You cut yourself. To stop the bleeding, first of all, the blood vessel constricts. That's vascular spasm. Second thing is because you've cut the vessel, there's all these free collagen fibers. Now, these free collagen fibers are sticky, and platelet comes along and sticks to it. More platelets, more stickiness. More platelets, more stickiness. Forms a little platelet plug, further stopping bleeding. Platelets will release a whole bunch of chemicals that trigger fibrinogen to turn to fibrin, which are fine fibers that then form a final clot. That's hemostasis. Make sure you know that. All right, blood groups. 4 blood groups. What do you need to know? Remember, you're going to have A, B, and O. A can be A positive or it can be A negative. B can be B positive or B negative. O can be O positive or O negative, but you can also have AB positive and AB negative. You need to know who you can donate blood to, who you can accept blood from. There's definitely questions in the exam asking you if you're, for example, AB positive, who can you give blood to? Well, who else can accept that? The way you remember it is this. If I give you a question saying somebody is AB positive, who can they give blood to? Just remember, if you're AB positive, you will create antibodies against everything you are not. If you're AB positive, there's nothing you don't have, which means you can accept everyone's blood because your body will not react to it, all right? But if you're AB positive and you're delivering blood to somebody, the only other person that can take your blood is another AB positive person, because if they're anything else, they're going to create antibodies against you, all right? So just remember that, work it through like that. Describe the structural and functional aspects of lymph nodes, and lymph organs. Main thing you need to know here is the difference between primary and secondary lymph organs. Primary versus secondary. Primary lymph organs is going to be the thymus, and it's going to be the bone marrow. That's it. Thymus creates T cells, bone marrow creates B cells. Secondary lymph organs include the lymph nodes, include the spleen, include the malt patches within our intestines, include our tonsils. These are all secondary lymphatic structures. Know the difference. All right, innate versus adaptive immunity. The difference between innate versus adaptive. Innate doesn't change. Doesn't change its response. It's always the same. Adaptive changes depending on what's invading. Adaptive predominantly is going to be the B cells and the T cells. The innate is going to be things like inflammation, fever, skin, mucus, some basic chemicals, some cells like macrophages. Doesn't matter what's invading you, these will always be released and do the exact same thing. So it's innate. Adaptive, they get released depending on what's coming in. Now, B cells turn into plasma cells, and they turn into antibodies, right? T cells, you can have helper cells or cytotoxic. Simply put, bacteria is invading. This will do whatever it can. If it doesn't work, at least the macrophages will have engulfed some bacteria, release some antigens. These antigens will be picked up by the helper T cells. They'll call in the B cells, which will turn into plasma cells, produce antibodies specifically against that bacteria or virus, right? The cytotoxic T cells then will go along and find any antibodies that are bound to any of these bacteria viruses and just kill them off straight away. All right. And then the last thing you need to know before my voice falls away and disappears is this. You need to know the difference between passive and active immunity. All right. Passive immunity. Okay, let's do active first. It's easy. Active immunity is when you create the antibodies against antigens you've been exposed to. Passive is you receive the antibodies from somebody else who has already made them. Someone or something else. All right? So, passive, you're receiving the antibodies that have already been made. Active is you're creating the antibodies yourself after exposure. And there's different types. There's artificial and natural, right? So natural passive will be like a baby with breast milk. They're getting antibodies that their mum's made. Artificial is going to be when you inject serum into somebody that contains antibodies. It's not that common, but it happens with those who are immunocompromised. When you can't expose them to the antigen, they may get even more sick. So you give them the serum, doesn't last very long, doesn't have a lifetime immunity. Active also has natural. Natural is when you get the flu, right? Someone sneezes in your face and exposes you to the antigen, you create the antibodies. Then you've got the artificial. This is just vaccines. All right, I'm going to have to stop because I'm losing my voice. These are the learning outcomes. Thank you, everybody. I'm sorry it took so long, but there's a lot of learning outcomes for the course. Best of luck for the exam.